Hi there, welcome to Spec Point 3.9, which is all about meiosis. So, we all know that mitosis is how your body cells divide to make new body cells for growth and repair. Now, meiosis is different because meiosis is the process where cells divide to make gametes. Okay, now your gametes are the sperm and the egg. And there's a really significant difference between meiosis and mitosis is that in meiosis, these gametes are all genetically different. Okay, whereas in mitosis, it makes cells that are genetically identical. Meiosis makes daughter cells that are genetically different. Okay, so they're non-identical. The other thing is that they have half the number of chromosomes. So gametes, in humans anyway, have 23 chromosomes, and we call that haploid. So they're haploid cells because they only have one of each pair. Now, in a different species, it will be a different number. In humans, it's 23 chromosomes, which is only one of each pair, so it's called haploid cells that it's making. Now, specifically, this spec point wants you to know how we get this genetic variation. So there's two things you need to know. The first one is called independent assortment of homologous chromosomes, and the second one is called crossing over. So we'll go through each of those in turn. Now, in some books, you'll see the whole step-by-step process of meiosis, but you do not have to know that. That is not what they want you to know. They want you to know how you get genetic variation from meiosis, not the details of the stages of how it actually happens. The specification reads, understand the role of meiosis in ensuring genetic variation through the production of non-identical gametes as a consequence of independent assortment of chromosomes and crossing over of alleles between chromatids. And it even says in brackets, details of the stages of meiosis are not required. Okay, so you do not need to learn the prophase, metaphase, anaphase, telophase, etc., how that happens twice. You don't need to know all that. So the first way we get variation is this, independent assortment of homologous chromosomes. So in the stage which we call metaphase, and you know metaphase from your mitosis work, in metaphase, the chromosomes line up on the equator. Now, in mitosis, that's a single line of chromosomes, but in meiosis, they line up in pairs. Now, we know that we have 23 chromosomes in the daughter cells that we're going to make, but the cell that it comes from has 46. So it has the pairs of chromosomes, and the chromosomes line up in pairs. So you've got two chromosome number ones next to each other, two chromosome number twos next to each other, two chromosome number threes next to each other, etc., all the way up to pair 23. So you've got pair 1, 2, 3, 4, 5, all the way up to pair 23. So you'd have 23 pairs all along here. Now, each pair, if this is our pair of chromosome number 1, this one will have come from the mother and this one will have come from the father. So this one's the maternal chromosome. This is the paternal chromosome. Then chromosome number 2 will have one that's maternal and one that's from the father that's paternal. Okay, and then of chromosome number 3, we're going to have one from mother, one from father, a maternal one and a paternal one. Now, when they line up on the equator here, so we call the middle of the cell the equator, when they line up on the equator of the cell in metaphase in meiosis, they line up in their pairs. But the significant thing now is, where does the maternal one go and where does the paternal one go? So it might be that of this first pair here, we get the paternal one up here and the maternal one down here. And of the next pair, we might get the maternal one up there and we might get the paternal one down here. And the next ones might all be like this, and then the last one might be like that. So it's totally random how they line up. Now, they don't actually line up 1, 2, 3, 4. It might be 1, 13, 12, 2, 16, whatever. Okay, but they are in their homologous pairs. So the two chromosome number 1s are called a homologous pair. And that means that they have the same genes at the same loci. Okay, so within those two homologous chromosomes, whatever gene this one has just here, this one has it just here as well. Whatever gene it has just here, this one has it just here as well. Okay, so they have the same genes at the same loci. Now, the one from mum and the one from dad might have different alleles. So one might have like blue eye allele, one might have brown eye allele, but they're for the same gene. So the eye colour gene, hair colour gene, hair colour gene. Okay, but the way that the paternal and the maternal line up is completely random. So I'm just going to type that in there. So it's random lining up of maternal and paternal chromosome from each homologous pair. Yeah, make that a bit smaller. Oops, it's a bit small. Okay. So the next stage of this is they get pulled to the poles. So the ones on the top row are going to get pulled to one pole, and the ones on the bottom row are going to get pulled to the other pole. So at this pole, we're going to end up with that blue, and this one we're going to end up with these 4 blues, and this one's going to end up with these 4 reds in the middle, 4 maternals, and maternal there, maternal there. So these have moved now to the poles, so they're no longer on the equator. They're at the poles. And then in the final stages, what happens is this membrane nips in. Okay, it's more complicated than this. There's lots of stages, but ultimately what happens is those ones end up in one daughter cell, one gamete, and these end up in another daughter cell, another gamete. So these ones have got this mix of chromosomes, so 4 maternal and 2 paternal. This one's got 4 paternal and 2 maternal. So these, depending on which one of, say, their eggs, which one of those eggs gets fertilised, the baby is going to have different alleles, aren't they? This one's going to be more like mum, this one's going to be more like dad. So, independent assortment of homologous chromosomes is the random lining up of maternal and paternal chromosomes from each homologous pair on the equator, and then they get pulled to the poles to make different gametes. So the gametes are different. Crossing over happens also in metaphase. Now, if we imagine that in metaphase, when we're lining up on the equator like this, if we were to zoom in on 2 chromosomes that are right next to each other, I'm just going to draw them in different colours so you can see. Sometimes these bits get almost like tangled up and they actually snap off like this. And then they fix themselves, but they fix themselves, I'll just rub a bit out, they fix themselves sometimes correctly, but sometimes incorrectly. So sometimes they fix themselves. So this chromosome has a little bit of that one on it, and this one has a little bit of that one on them. Okay, so what happens is when the chromosomes line up on the equator, let's make that box a bit bigger. Break off and rejoin, sometimes swapping some of the genes. Okay, so what can happen then is that whichever gamete inherits this allele gets all the genes that are on in this chromosome, gets all the genes that were paternal, but also gets a little bit here, so it gets a few maternal genes. Okay, so it just makes it all a little bit more complicated and causes a little bit more variation, which is the whole point of meiosis, to get variation in all those alleles. So all the eggs from one person, all those egg cells are different, and also all the sperm cells are different. And the reason they're different is due to the way they line up in their pairs in independent assortment of homologous chromosomes, and crossing over when the chromosomes are close to one another, little bits snap off and they get mended and they fix onto the wrong chromosome. Okay, and that's all there is that you need to know about meiosis.